Hello students, I am Tulika Banerjee. Today, I bring you the next learning episode in BSc Forensic Science on an important unit of the paper titled Fingerprints, that is detection of blood prints. In this lecture, you will be introduced to the detection of blood prints. You will also be studying about blood smeared fingerprints and detection of blood fingerprints with heme-reacting reagents and detection of blood fingerprints with protein reacting reagents. We will wind up this module with a conclusion. Let us start our today's lecture with a brief look at what we will be learning today. First, we have the introduction to detection of blood prints. Then we have blood smeared fingerprints. The third module here is detection of blood fingerprints with heme reacting reagents, and the fourth one is detection of blood fingerprints with protein reacting reagents, and last is the conclusion. So, dear students, let us start with the introduction of this lecture to detection of blood prints. Latent prints are mostly encountered at the scene of crime. The investigators use the general technique of oblique lighting to locate these latent finger marks. After locating these prints, certain physical and chemical techniques are required for the development. The physical techniques generally include the powder method, whereas the chemical techniques include the application of certain chemicals such as ninhydrin, iodine, cyanoacrylate fuming, etc. There are a host of chemical techniques for detecting latent fingerprints on different surfaces such as iodine fuming, use of ninhydrin and its analogues, cyanoacrylate fuming technique, and many more. But however, if the surface happens to be stained by blood, as is common at crime scenes such as in case of murders and assault, then special procedures have to be adopted to lift the prints. The main problem encountered with blood prints is that not every chemical reagent will react with the heme component found in blood to give the prints. Moreover, the blood sample has to often be submitted to the Forensic Biology and Serology laboratory for further investigation. So, the process of investigating the fingerprint sample needs to be very precise manner. The blood stains are often found on weapons, victims' bodies and other articles removed from the scene of crime. A scrutiny of these items may also reveal bloody fingerprints. Such fingerprints are generally visible since the ridges become stained with the red color of blood. The image here shows how blood impressions are usually discernible. However, if a blood coated finger touches a surface repeatedly, the impression gets weaker and weaker, or it may get fainter or fainter. Under these circumstances, it is pertinent to enhance the impression with the aid of a suitable chemical reagent so as to make the ridge detail clearer and sharper. The chemical reagents which enhance the blood fingerprints often tend to destroy or weaken the enzymes of forensic interest. Therefore, prior to fingerprint detection, blood samples from the impression should be collected for serological examination. When fingerprints impinged on a blood-stained article are to be detected, the method of choice depends on the nature of the surface and, more importantly, on the state of the coagulated blood. The nature of surface also plays a very important role, because if it is an absorbent surface, the blood, rather than forming a print, will seep down into the surface, whereas clearer prints will be obtained on non-porous surfaces. The next factor is the state of the coagulated blood. On coagulation, the serum content of blood separates out from the cellular content. The blood cells usually form a solid red mass which is surrounded by a layer of straw colored serum fraction. The serum portion is rich in soluble proteins. The blood cell fraction is rich in heme component. There are two broad methods for detecting blood fingerprints. If the finger touches predominantly the red blood cells on the surface of the crime scene evidence, the print may be developed by the application of a reagent that reacts with heme content of coagulated blood. If, on the other hand, the finger comes into contact with a portion of crime scene evidence which is coated with serum, the print may be developed by the application of a reagent that reacts with the protein content of coagulated blood. The heme moiety of the hemoglobin in blood catalyzes the oxidation of a number of organic reagents. The oxidized reaction product is normally colored and therefore renders visibility to fingerprints. Prior to 1974, benzidine was the most popular heme-reacting chemical used for detecting blood fingerprints. The image here shown on your screen represents the structure of benzidine. Subsequently, however, benzidine was found to be carcinogenic in nature and the fear of occupational exposure or occupational hazard prompted the health authorities to ban its use as a fingerprint reagent. Dear students, in this section I will now be taking up the heme-reacting reagents that have proved useful and safe for detection of blood fingerprints. So, here in this section the very first reagent is 3,3-5,5-tetramethylbenzidine. The chemical reagent 3,3-5,5-tetramethylbenzidine, the structure of which is shown on your screen, is non-toxic and very effective in detecting blood finger marks. Now, how this solution is prepared? A buffer solution containing 10% by weight of sodium acetate and 80% by volume of glacial acetic acid is prepared. To 100 ml of this solution, 2 grams of 3,3-5,5-tetramethylbenzidine reagent is added. This is marked as solution A. A mixture of colloid around 30 ml, ethanol around 15 ml and diethyl ether around 120 ml is separately prepared and this solution is marked as solution B. So, we have two solutions, solution A and solution B. The working solution is prepared by dissolving 6 ml of solution A and 0.5 grams of sodium perborate in 120 milliliters of the solution B. The surface is impinged 2 to 3 times with the working solution from a distance of about 6 inches using a spray gun, one variety of which is shown on your screen. On drying it at room temperature, the finger marks are rendered visible. The second chemical reagent that reacts with the heme component of blood is phenolphthalein. A mixture of phenolphthalein around 2 grams and whose structure has been shown on your screen is taken. Then potassium hydroxide around 20 grams is taken and powder zinc, that is 20 grams, is placed in distilled water around 100 ml. The contents are refluxed for 2 to 3 hours until the formulation becomes colorless. This solution is kept in a dark colored bottle and care is taken that it remains in contact with zinc powder. Immediately before the fingerprints are to be developed, 20 ml of this solution is mixed with 80 ml of ethanol and 0.5 ml of 3% hydrogen peroxide. The test solution is applied to the article containing blood prints. On drying, the fingerprints are rendered visible or they become visible. The third type of reagent is leucomalachite green. An acidic solution of leucomalachite green reagent, the structure of which has been displayed on your screen, is quite effective in developing blood fingerprints or blood finger marks. How this solution is prepared? To a mixture of diethyl ether around 70 ml and glacial acetic acid around 1 ml, leucomalachite green around 1 gram and hydrogen peroxide 20%, 0.5 ml are taken and added. This working solution is sprayed onto the surface impinged with blood finger marks. On drying, the fingerprints are visualized. Detection of the blood fingerprints with protein reacting reagents. There are a good number of organic derivatives which bind to the protein moiety in the blood to yield colored complexes. However, blood proteins are soluble in water. Therefore, before the article under examination is dipped in the test solution, it is baked in an oven for appropriate time, depending on the reagent that is being used on that particular surface. As a result, the proteins get denatured and become fixed to the surface. Thereafter, the item is sprayed with the test solution of the reagent. The following protein reacting reagents have proved efficient in detecting blood fingerprints. The very first reagent which reacts with the protein moiety is AMIDO black. While using AMIDO black reagent, the structure of which has been shown on your screen already, 3 solutions need to be prepared. Solution A, or the very first solution, contains AMIDO black 0.2 grams dissolved in a mixture of methanol, that is 90 ml, and glacial acetic acid around 10 ml. Solution B, or the second solution, is a mixture of glacial acetic acid taken 10 ml and methanol taken 90 ml. The third solution, or solution C, also contains a mixture of glacial acetic acid around 5 ml and methanol around 90 ml, but in a different proportion. The surface to be examined is baked in an oven at 100 degrees centigrade for about 30 minutes. It is then immersed successively in solution A and B. Finally, it is washed with solution C. The surface is allowed to dry naturally, whereupon the fingerprints are rendered visible. A representative blood fingerprint developed by AMIDO black reagent has been shown on your screen. The second chemical reagent that reacts with the protein component is crystal violet. The working solution is prepared by dissolving 0.1 grams of crystal violet, the structure of which is shown in this figure, in 100 ml of distilled water. The pH of the solution is adjusted to nearly 8 by adding a few drops of 1:1 aqueous ammonia. The article to be examined is heated to around 100 degrees centigrade for about 30 minutes. It is then sprayed with the test solution. After 2 to 3 minutes, it is rinsed with distilled water. On drying, the finger marks are rendered visible. A sample blood fingerprint developed by crystal violet reagent has been shown on your screen. The third chemical reagent here is Kumasi Brilliant Blue R. 2 solutions are required for developing blood fingerprints with Kumasi Blue reagent. The staining solution is prepared by dissolving 2 grams of Kumasi Brilliant Blue R 250 dye in a mixture of glacial acetic acid taken 20 ml and methanol taken 100 ml. The de-staining solution contains all the ingredients in the same proportion of staining solution except the dye. The structure of Kumasi Brilliant Blue R has been shown on your screen. The article to be examined is baked at 100 degrees centigrade for 30 minutes. It is then immersed in a staining solution for 3 minutes and subsequently rinsed with the de-staining solution. On drying, the finger marks are rendered visible. Kumasi blue reagent may also be used in concert with cyanoacrylate method of fingerprint detection. Cyanoacrylate method is based on the principle that when alkyl 2-cyanoacrylate is allowed to vaporize, it undergoes polymerization. The polymerized ester has a tendency to get adsorbed on the sweat residue, imparting a white color to the ridge pattern. The color contrast may be improved by various post-treatment methods. The surface impinged with blood prints is first baked in an oven for 5 minutes. It is then suspended from the roof of a cabinet containing a china dish comprising of cyanoacrylate ester. The item is exposed to the vapors for about 2 hours until whitish colored fingerprint pattern develops. Thereafter, it is successively treated with the Kumasi Brilliant Blue R staining and de-staining solutions. On drying, fingerprints appear. Next reagent is the Crowell's reagent. Crowell's reagent is prepared by dissolving a mixture of Kumasi Brilliant Blue R dye taken around 0.15 grams and Crocine Scarlet 7B taken around 2.5 grams, the image of which is shown on your screen. In glacial acetic acid at around 30 ml, trichloroacetic acid around 30 ml and distilled water around 920 ml. After the article is baked at 100 degrees Celsius for 30 minutes, it is immersed in the test solution for about 5 minutes with constant agitation. It is then repeatedly washed with water until the test solution background coloration disappears. The surface is then allowed to dry, upon which the fingerprints appear. The next reagent is ninhydrin. Ninhydrin reagent or ninhydrin technique has traditionally been the most popular one for processing latent fingerprints on porous absorbent surfaces like paper, cardboard and wood. The method relies on the reaction of ninhydrin, the structure of which has been depicted on your screen, with amino acid content of the fingerprint residue. The reaction produces a purple colored compound called as Raman's purple, as is shown in the figure on your screen, which becomes deposited along the ridges, making the latent prints visible or rendering them visibility. A 0.2 to 1.5% solution of ninhydrin in Freon 113 is sprayed on the surface containing the finger mark from a distance of about 6 inches. After the solvent evaporates, the solution is re-sprayed. The surface is then heated to about 80 degrees centigrade without allowing it to come in contact with the heat source. Better results are obtained by steaming the article. Optimum development of fingerprints occurs at a relative humidity of 65 to 80%. Ninhydrin method makes it possible to develop fingerprints that are many years old. However, since the concentration of amino acids in perspiration is usually quite low, the developed prints normally do not show a sharp contrast. In order to solve this problem, the developed prints are post-treated with a group 12 metal salt, generally zinc chloride. This interaction converts the non-fluorescent Reagents purple into a luminescent complex, the structure of which has been depicted on your screen. The fingerprints developed by ninhydrin zinc chloride method become highly fluorescent under argon laser light. This procedure may be extended for developing bloody fingerprints also. The blood protein is denatured and fixed on the surface by baking the sample in an oven at 100 degrees centigrade for about 5 minutes. Thereafter, the prints are sprayed with ninhydrin, followed by zinc chloride treatment and then examined under laser. Now, dear students, let us conclude this lecture. Latent prints are mostly encountered at the scene of crime. The investigators use the general technique of oblique lighting to locate these latent finger marks. There are a host of chemical techniques for detecting latent fingerprints on different surfaces, such as iodine fuming method, use of ninhydrin and its analogues, cyanoacrylate fuming technique and many more. However, if the surface happens to be stained by blood, as is common at crime scenes, then special procedures have to be adopted to lift the prints. There are two types of chemical reagents which react with blood. One type of reagent reacts with the heme component, whereas the other type or the second type reacts with the protein component. With all these information, here we come to the end of today's lecture. Do keep in mind what we have discussed today. I will be back with one more lecture in this series. If you want to learn more and enhance your knowledge, you may log on to our website www.cec.nic.in for MCQ, quizzes, LORs, etc. Thank you for your time today. I will see you in the next lecture. Have a great day.